Welcome everybody to our Immunology Pills mini webinar series with today's teaser presentation, Dendritic Cell Research Solutions. In the next 20 minutes, you will learn about Miltenyi tools and methods for studying dendritic cell subpopulations in human and mouse. For deeper and more extensive information, please contact your local sales organization and visit our website. The disclaimer is displayed here. And our agenda for today. We start with a brief introduction. Next, we'll talk about sample prep for dendritic cells. Then flow cytometric analysis of human and mouse dendritic cells. We'll then tell you how to isolate dendritic cells in a simple way. And finally, we'll describe the workflows for MoDC generation. Introduction. Dendritic cells were discovered by Paul Langerhans in 1868. Their function was described by Rolf Steinman in 1973. Dendritic cells are professional antigen-presenting cells that promote immunity to foreign antigens on one hand and induce tolerance to self-antigens on the other. They regulate the immune response by secreting pro- and anti-inflammatory cytokines. And these cells come in many flavors, as can be differentiated based on their expression of cell surface markers, tissue localization, ontogeny, and their functional specialization. In a very simplified view, in both human and mouse, three major dendritic cell subsets can be found. Myeloid, conventional dendritic cell 1, myeloid or conventional DC2, and plasmacytic dendritic cells. Depicted here, monocyte-derived disease do not share ontogeny with conventional dendritic cells and can be derived from monocyte lineage during infection and inflammation. Due to time constraints, we'll not be discussing further subsets of dendritic cells, but I encourage you to stay tuned and learn more, for example, from publication list shown here and from published scientific papers. As mentioned before, these are specialized antigen-presenting cells that play a key role in initiating and regulating innate and adaptive immune responses. But these cells are fundamentally different in many ways. Here, we'll focus on key characteristics of human dendritic cells. Diseases have different phenotypical features and express various sets of markers that can be used to identify those cells. Among the most commonly used markers, CD141 expression is mentioned in the context of DC1, CD1c is expressed by DC2, and PDCs express CD123, CD303, and CD304. These C subsets have different ontogeny and depend on different growth factors. Their identity is regulated via expression of various transcription factors, and functional specialization of these C subsets results from the expression of receptors, including pattern recognition receptors, but also various cytokine secretion abilities. Finally, there is an obvious division of labor between DC subsets where DC1 are potent inducers of Th1 immunity, DC2 can induce Th1, Th2, and Th17 responses, and PDCs mount antiviral responses. Now, the sample preparation for dendritic cell analysis. Preparing samples for DC analysis can be overwhelming because there are many tissue sources and different labs use different protocols. In laboratories working with human dendritic cells, the most commonly used tissues are blood and blood products such as buffy coats, LRSCs, and leukapheresis. For mice, blood samples are also collected. Here, sample preparation for analysis is easy, and researchers often perform density gradient centrifugation or RBC lysis for both human and mouse. Human bone marrow samples are usually purified by density gradient centrifugation and RBC lysis, whereas in mice, investigators usually harvest femur and tibias and flash the bone marrow with medium or buffer. The analysis of tissue resident or infiltrating dendritic cells is more complicated due to tissue-specific enzymatic digestion and dissociation protocols. Miltenyi Biotec engineers, in close collaboration with researchers, have developed GentleMax tissue dissociators, GentleMax tubes, and tissue dissociation kits to simplify the labor-intensive process of preparing single-cell suspension for DC analysis, with a focus on cell viability and epitope preservation. Here, an exemplary analysis of mouse disease subsets after GentleMax dissociation of spleen, lung, and lamina propria is shown. Enzymatic tissue dissociation with GentleMax is a gentle method to obtain a single-cell suspension for further analysis. The enzymes included in Miltenyi Biotec kits are highly purified to maximize epitope preservation and the composition of digestion medium, but also the mechanical dissociation parameters are optimized to ensure high cell viability. Lot-to-lot consistent enzymatic activities, automation, and standardization ensure high intra- and interlaboratory reproducibility. Flow cytometric analysis of human and mouse disease. Flow cytometry is a mainstream tool for studying dendritic cells. The best tool Miltenyi Biotec has to offer to anyone trying to accurately analyze cells by flow cytometry is REAfinity antibodies, which are recombinantly generated antibodies in which the Fc region has been mutated in order to eliminate background signal. Translated into myeloid language, the FcR blocking step is not needed. REAfinity antibodies show high epitope specificities, low background binding, are all of human IgG1 isotype, making isotype control staining easy, and overall they ensure consistent and reproducible experimental results. Once we have selected the reliable antibodies for DC staining, we need to decide on the specificity of the antibodies, therefore the markers we want to stain on our cells, and the dyes and fluorochromes. The table on the left-hand side shows an optimized panel for analysis of splenic disease using MaxQuant or similar flow cytometric analyzer. The gating strategy starts with selection of cells and exclusion of debris using the side and forward scatter plots. Next, we select singlets and then live cells where viability dyes such as propylium iodide or 7-AD can be used. Whenever necessary, we recommend macrophage exclusion using, for example, F480 and CD64 staining. And finally, we can analyze the abundance of DC subsets in mouse spleen. Conventional DCs show high level of CD11c and MHC class II expression and can be divided into two major subsets, XER1 expressing DC1 and CERP alpha or 11b expressing DC2. Plasmacytic disease usually express intermediate level of CD11c, therefore Siglec H and PDC1 staining is recommended to facilitate more precise gating. Similar to described on the previous slide, splenic disease staining and analysis procedure, the cells shown here were stained with the antibodies described in the panel on the right-hand side. After exclusion of dead cells and debris, CD11c MHC class II positive conventional dendritic cells were selected and further subdivided into XCR1 and CD172 or 11b expressing subsets. Plasmacytic dendritic cells can be easily identified by cyclic H and PDC1 expression. You could find a detailed staining protocol under the link provided here on the Miltenyi website once you open the PDF file of this presentation. In order to analyze the abundance of the three main DC subsets in human blood, we have developed a staining panel and a gating strategy in collaboration with Yolanda de Vries from the Nijmegen University. After selecting viable CD45 positive cells and excluding B cells and monocytes, the three DC subsets can be identified, where cross-presenting DC1 co-express CLEC9A and CD141, DC2 are Fc epsilon receptor 1 alpha positive and CD1c positive, and plasmacytic DCs express CD123 and CD303. The outcome of such staining is very nicely depicted on the dot plots on the right-hand side. This slide demonstrates a detailed step-by-step gating strategy in order to evaluate DC abundance in blood samples. Cells from lysed blood were stained using the panel shown on the left, and for the gating strategy, first debris and then 7AD positive dead cells were excluded. Next, CD45 positive cells were selected, and from there, side scatter low cells were gated. CD14, CD20 negative cells were selected, and within these, the abundance of PDCs expressing CD123 and CD303, as well as CD141 expressing CDC1 was evaluated. Next, after excluding CD123 positive cells, the abundance of Fc epsilon receptor 1 alpha, CD1c, double positive DC2 was assessed. You could find a detailed staining protocol under the link provided here on Miltenyi Biotec website. Now, the magnetic isolation of dendritic cells. In order to perform molecular analysis of cells or cell culture experiments, researchers need to isolate cells of interest, and there are many methods available to isolate subsets of dendritic cells. Here, we'll focus on the easiest and simplest cell sorting method that does not require expensive equipment: magnetic separation using MAX technology. In addition of being a simple and cost-effective method of cell purification, MAX separation allows us to obtain high-purity cells for downstream applications. This method significantly reduces the time required for flow cytometric analysis of rare DC subsets and can reduce the time and costs needed to conduct cell sorting, as pre-enriched cells will be present at higher frequency in the cell suspension from which we want to isolate the target population. Depicted here is a dot plot showing low abundance of resident and migratory disease in mouse lymph node cell suspension, where more than 60-fold enrichment of target cells was achieved using CD11c MicroBeads UltraPure. Finally, MAX beads are compatible with most laboratory techniques and applications. The CD11c MicroBeads UltraPure mentioned on the previous slide were used to isolate CD11c positive cells from spleen, lung, lamina propria, and tumor. Tissues were harvested and dissociated using tissue-specific dissociation kits and gentle MAX dissociator. The cell suspension was then labeled with CD11c MicroBeads UltraPure and magnetically separated, followed by flow cytometric analysis to compare the abundance of disease subsets in the starting material and the enriched fraction, as depicted on the upper panel. The bottom panel beautifully shows 10 to 42-fold enrichment of disease in tissues tested, which facilitates the use of beads to isolate disease or to increase the frequency of these cells and reduce the time required for flow cytometric analysis or sorting of disease subsets. Miltenyi Biotec has many products and solutions for optimal enrichment of mouse disease. Visit our website or contact your local sales representative to find a product tailored to your needs. When working with human disease, you definitely try to find an easy protocol to isolate disease subsets. Here we show an example of enrichment of human cross-presenting CD141 positive disease. Cells isolated with microbeads are highly enriched and express DC1 surface markers such as CLEC9A, CD141, but also HLA-DR. Upon stimulation with toll-like receptor 3 agonist poly-IC, CD141 disease upregulate the expression of CD80, 83, CD86, CCR7, HLA-ABC, and HLA-DR, but also secrete cytokines such as interferon alpha, IL-2, IL-4, IL-6, TNF-alpha, and interferon. In this experiment, cross-presenting disease were isolated from cytomegalovirus seropositive donors using CD141 microbeads. Enriched cross-presenting disease were incubated with PP65 and IE1-derived peptide pools and simultaneously activated with poly-IC for 6 hours. Autologous CD8 positive T cells were isolated using the CD8 T cell isolation kit and labeled with cell tracer. After 10 days, T cell proliferation was assessed. Antigen-loaded CD141 disease effectively cross-presented antigens and induced antigen-specific T cell proliferation in cell culture, what can be concluded from the reduced cell tracer intensity compared to the control disease sample. Furthermore, the abundance of CMV-specific CD8 T cells was also detected by pentamer staining. For more details on this experiment, please see the poster on Miltenyi website. Miltenyi has many solutions for isolating human disease. As before, you could visit our website or contact your local sales representative to find the product that fits your needs. And finally, the MODC generation part. Generation of MODCs begins with the isolation of monocytes. The solutions presented here are developed to facilitate the isolation of total or pan-monocytes, CD14 positive classical monocytes, and CD16 positive, CD14 negative non-classical monocytes. However, for MODC generation, researchers use either CD14 positive classical monocytes or pan-monocytes. Here, CD14 microbeads are the most commonly mentioned and cited solution. The product highlight: isolation of CD14 monocytes can be performed directly from buffy coats, LRCs, or leukoplaques. Here, Miltenyi Biotec offers StraightFrom CD14 microbeads to facilitate time-efficient monocyte isolation. Finally, the complete workflow for MODC generation. First, isolated monocytes are seeded at the density of 1 million cells per ml of complete medium containing 1% autologous plasma and supplemented with max premium grade cytokines, IL-4 and GM-CSF. Typically, each second or third day, a portion of exhausted medium is replaced with a fresh medium supplemented with cytokines. On day 6, immature modices can be stimulated with, for example, inflammatory cytokines such as TNF-alpha, IL-6, IL-1 beta, or toll-like receptor agonists such as LPS. During stimulation, disease may also be posed with antigens in order to assess the ability of activated modices to present antigens to T cells. This figure shows changes in surface marker expression at different stages of modice generation, highlighting the typical DC phenotype obtained by modices generated in vitro. The figure in the middle panel shows the migration of mature CCR7 positive modices towards the CCL19 chemokine gradient. And here, ineffective IL-12 production and low IL-10 production by mature modices after stimulation with CD40 ligand is highlighted. A detailed modice generation protocol and additional details for downstream applications can be found at the link provided on the slide. Our goal at Miltenyi is to support your DC research with quality products, verified protocols, and application notes. And of course, you could always directly reach to our local sales organization for deeper and more extensive information. Here, I would like to thank you for your attention.